We talk about the fact that we all have stem cells in every organ or tissue, and it's true, we do. But one thing that very few people talk about is the fact that, and one of the things that we're working on is the fact that as we age, we lose some of our stem cells. So I actually believe that aging is a failure of stem cells. What do I mean by that? What I mean is that as we get older, not only do the number of cells that we have that can repair our body go down, but the function, the ability of the ones we have goes down as well. You know when you're 2 and you fall down and you scrape your knee? You heal and you don't have a scar for the rest of your life. When you're 62 and you fall down and you scrape your knee, you're probably going to have a scar. And that's because as we age, we lose stem cells, cells that our body uses to repair every organ or tissue. The cool thing is, and not many people are looking at this, is there's a difference in how that happens in men and women. So one of the things that I'm thrilled about is the chance to work with Stephanie Coulter at the Women's Heart Health Program here, along with the colleagues I'm already working with around the country, to begin to define the differences in how men and women repair their bodies, how our stem cells work, and how they differ in men and women. You know, we know women rock. Now we've got the data to prove it.
So, shall we have a look at your baby? You're 27 weeks now, aren't you?
Yeah.
Okay. Listen to baby's heart beating. Perhaps a little bit. Oh, that's a nice one, isn't it? It's always good, isn't it?
Birth defects represent one of the major cause of morbidity and mortality in children. And there's about 3 every 100 babies which are born with a major birth defect. We are facing many times with a situation in which we don't know what to do for these babies.
It's a girl. Okay. I collaborate with Paolo Di Coppi. I look after the mums while they're pregnant. Paolo visits the fetal medicine unit and he sees the mums if the baby has an abnormality because he looks after the baby when they're born. And we're trying to develop treatments using stem cells in the amniotic fluid around the baby.
The stem cells have represented a major breakthrough because it's the possibility of growing cells outside the body to make a repair to the children once the cells have been built in the laboratory. So what we have developed in the last few years, and this has been initially the work we have done with Anthony Atala at the Wake Forest Institute of Regenerative Medicine, and is the possibility of deriving stem cells from the amniotic fluid. These are not embryonic stem cells, but are neither adult stem cells. They have characteristics that are in between the 2 cell types, and for that they may represent a big advantage for therapy. These cells would represent an ideal source for building organ and tissue that are missing in the fetus. And this is because we can predict and diagnose very accurately these diseases before the fetus is born. Once this diagnosis is made, however, we have about 20 weeks of gestation in which we can plan the engineered organ to be built outside the baby, that then can be eventually implanted once the baby is born. So we can correct that defect at birth using its own cells. Here is where we do receive the samples from Anna's. Once the cells are isolated from the amniotic fluid, they can be easily expanded in the incubator. and eventually engineer in a three-dimensional structure that can mimic the organs that the baby is missing. These organs can be expanded and grow in these bioreactors.
We haven't got any treatments that will actually work yet, but we're working on this whole area. For instance, the mum that we scanned earlier on has got a completely healthy, normal baby as far as I can tell on scan, and we're not looking to treat anybody right now, but we are trying to develop new treatments which will improve already existing treatments or to develop completely new therapies using the stem cells that are in the amniotic fluid.
In other situations in which we can intervene even before birth, we can use the same cells to treat the fetus. So if we know that the fetus has some malformation that can be corrected before birth, we can use his own cells for a treatment. So we can culture his or her cells outside the womb and expand them and eventually correct, for example, a gene that is altered or missing using gene therapy technique, and we can inject back the cells into the fetus before birth, and so somehow improving his option of life once the baby is born.
As humans, we develop from a group of cells called embryonic stem cells. These cells can make up all of the tissues in our bodies. But once we are born, these embryonic stem cells are long gone. In place of these cells, our tissues have their own specialized stem cells that can repair that specific tissue in case of injury, or to replace cells that are worn out. But they can only produce cells of that tissue and no other tissue. For example, a muscle stem cell can only produce a muscle cell, not a heart cell. That's the job of the cardiac stem cells. The scientists of this paper wondered if there may be some cells hiding out somewhere after birth that can make a whole range of tissues, like embryonic stem cells. This would be a very interesting find because of the ethical issues associated with studying and culturing human embryonic stem cells. But if there existed cells that resembled embryonic stem cells that could produce a variety of different tissues, it would open up many opportunities for regenerating entire organs made with a person's own cells, eliminating many complicated aspects associated with transplants, artificial organs, etc. These scientists are the members of the Tlisty Lab at the University of California in San Francisco, and their findings were published in their paper titled Rare Somatic Cells from Human Breast Tissue Exhibit Extensive Lineage Plasticity, published in the Proceedings, the National Academy of Sciences in March 2013. These scientists were studying normal human breast tissue and were interested in seeing if this tissue contained cells that could produce a variety of different cells from different tissues. To tackle this question, they looked at markers that are located on the surface of the cell that they found to be different between cells that had the ability to produce cells of different organs compared to cells that did not have that ability, but instead could only produce one type of cell. They found that two cell markers were different between these two types of cells. One marker was increased, CD73, and one marker was decreased, CD90. In cells that could produce cells of different tissues compared to cells that could not. This finding gave the scientists the ability to go on a fishing expedition to find cells in normal human breasts that had lots of CD73 and very little CD90. And surprisingly, they found some. Let's color these newly discovered cells in red from here on. They isolated these red cells and wanted to examine them a little closer. To start off, the scientists found that these cells contain a lot of the same proteins as embryonic stem cells. So they wondered if they were also able to form a variety of different tissues, like embryonic stem cells. So first, the scientists wanted to see if these red cells could produce cells of the breast tissue, where they were discovered. And indeed, when a group of red cells were allowed to divide in the presence of very specific conditions, the scientists observed the presence of normal breast cells in a petri dish. including the muscle cells that surround the mammary glands. But the scientists took this a step further and injected these normal breast cells in a mouse in which the breast tissue had been removed. And they found that these normal breast cells, produced by the red cells, could repopulate the mammary gland and even produce milk when the mouse was pregnant. So that's great. The scientists have discovered a stem cell that lives in the breast that can make breast tissue. But can it also make other tissues? Well, it turns out that they can. The scientists were able to make cells of the pancreas, lipid-filled fat cells, cells that line blood vessels, and even uniformly beating heart cells in a petri dish. To confirm that all of these different types of cells could be produced from one red cell, instead of many red cells that can each make one type of tissue, they took single cells, allowed them to divide, and then did the same assays, looking for their ability to produce breast, pancreatic, fat, blood vessel, and heart cells. And the scientists found that a single red cell could produce all of these tissue cells. So all of this data shows that these red cells, isolated from normal breast tissue from multiple women, have the ability to produce all kinds of different cells that are perfectly functional. So next, the scientists were curious to see if these red cells, like embryonic stem cells, could divide forever, which could suggest that they may be on their way to becoming cancer cells and could make it more difficult to use these cells for therapy. But surprisingly, the scientists found that these red cells could not divide forever. They had a very limited lifespan, unlike embryonic stem cells and many cancer cells as well. They also had perfectly normal chromosomes or DNA profiles. So these two aspects of their biology suggest that they are probably not precancerous cells that tend to have a lot of changes at the level of the DNA. So what did this study show? This study found two markers that could differentiate a cell that has the ability to make different tissues from a cell that does not: high CD73 and low CD90. The scientists call these cells endogenous pluripotent somatic cells, or EPS cells, which just means that they are found in our bodies, that they have the ability to make different tissues, and that they are not germ cells, which are eggs and sperm. By isolating and studying these EPS cells, the scientists found some similarities to embryonic stem cells. A single EPS cell can produce cells of all kinds of different tissues: breast, digestive system, heart, blood vessels, and these EPS cells were genetically stable, unlike cancer cells. But, contrary to embryonic stem cells, these EPS cells could not live forever, which, together with their stable genetic makeup, limits their ability to turn into a cancer. So what does this mean for you? This study is very significant for the world of regenerative medicine, since it provides a way to use a patient's own EPS cells to produce organs needed by that patient, which could potentially be transplanted back into the patient with low immune rejection, unlike traditional transplants, and possibly low cancer risk. Of course, much needs to be done before we can get to that point. Scientists are going to have to figure out if other tissues also contain these red EPS cells, understand their role in normal biology, and study how best to turn these EPS cells into a desired cell or tissue to treat a specific disease. No one thought these EPS cells could ever exist in the body. Why would we want cells that can make pancreas in the breast? But crazy enough, it turns out that there may be previous pathological evidence for their existence. Some pathologists have previously identified cases in which normal tissues grow in abnormal locations. For example, bone growing in the colon or the eye, liver growing in the gallbladder, or pancreas in the brain. This suggests that these EPS cells may be present all over the body. So as you can see, there's still much biology that we don't understand because of the difficulty in identifying and isolating rare cells. But with all of the new technology around, it's becoming much easier to do this kind of research. So scientists are now able to go back, ask new basic questions that they had always wondered about, and have the tools necessary to get clear and important answers like never before. So simple questions that seemed impossible to answer 10 years ago can now be answered and give us powerful new ways to rethink the way we treat disease.
It's a very exciting time. The next 50 years will be the golden age of cardiovascular disease. The opportunities to prevent heart and vascular disease, to make people's lives who have heart and vascular disease better and easier, and to correct the ravages of heart and vascular disease by regenerating hearts in their entirety are enormous.
As a patient said to me, we're not building organs, we're building hope. It's phenomenal to be able to imagine a time when we can take our knowledge of stem cells, our knowledge of organs, and build solutions for people with heart disease. Truly rebuild a functioning heart that we can transplant, so that we're not only the creators of the total artificial heart, but the creators of the total bio-artificial heart that lets you live the rest of your life with the solution built at THI.
The progress that is being made in the field of prevention of a stroke and treatment of common arrhythmias, with the revolution that was made by catheter ablation techniques, is exciting. The progress will help patients have safer, faster, and more curative results from the procedure that we do every day.
Valve disease is a product of aging, and the American population gets older every year, so that we are seeing more and more valve disease with each passing year. At the same time, we now have the ability to replace heart valves through the skin, as opposed to requiring heart surgery, at least in some cases. For all of those reasons, this is an extremely exciting time to be involved in the treatment of valvular heart disease.
We're really excited about the prospect of stem cell therapy for treating patients with heart failure as well as other cardiovascular diseases. One extremely exciting development is that this year in the third quarter, the Heart Institute will be leading a worldwide study to evaluate the effect of stem cells in patients with heart failure. This is building on our prior experience, and if this trial shows what previous trials have shown, this will ultimately lead to approval of therapy in the United States and in Europe.
See, the future today is that within the next few years, maybe even a couple of years, it will be possible to have every single individual's genetic information at the bedside. And that will be hopefully utilized by clinicians who have expertise in this area to benefit of the patient in their care, in their diagnosis, in their identification of those who respond better to therapy, in identification of those who potentially might develop the risk of side effects of the drugs, and hopefully therefore better treatment, better diagnosis, and more specific treatment of individuals and better outcome. This is the most exciting part of genetic research today, and we hope that tomorrow that will be at the bedside, applicable to every single patient.
Heart disease and cancer often coexist in the same patient. It is not widely known that cancer therapy can cause heart problem. We have identified the causes of cancer therapy-induced cardiovascular complications. In the future, we'll be able to predict and prevent and treat cancer patients that are undergoing a therapy to protect their heart so they can enjoy their life after cancer therapy.
Women's Center is actually beyond need at this point, as women, as you know, are dying in greater numbers than men. And I think it's time that we step up to the plate and address the needs and concerns of women.
Our ultimate goal is to develop a device that we could take off the shelf and implant in these gravely ill patients with heart failure that would not only prolong their lives, but restore them to a full and active lifestyle. Because there are 6 million Americans that have heart failure and 350,000 of them die every year. So it's a huge unmet clinical need. We're addressing this with a number of very innovative programs right here at the Texas Heart Institute. In fact, one device developed in this lab has already been used to save thousands of lives. And the progress we're making toward the development of the first fully implantable continuous flow artificial heart shows incredible promise. And Dr. Frazier has devoted his entire professional career to the development of these devices, and under his leadership, we'll continue to make progress in the war against heart disease. I feel so very fortunate and so proud to be part of this team.
The next 50 years will be very exciting, perhaps even more exciting than the last 50 years, because the genes will be identified and the most dangerous ones knocked out. We'll regenerate the whole heart from stem cells. We'll develop the ability to predict heart attacks in advance of their occurrence by identifying plaques in arteries that are dangerous even if they don't lead to symptoms. And we'll have brand new ways to treat every imaginable cardiovascular disease until it's gone.
I'd encourage everybody to read and learn as much about it as you can, because these people up here are doing a fantastic job.
This device has kept Mr. Craig Lewis, a 55-year-old man with cardiac amyloidosis who had his heart removed and replaced by these two pumps, alive now for 12 days and continuing to do better.
I recall 42 years ago when we attempted to use a total artificial heart for the first time in a human patient. And this initial success there was, I think, a milestone. For the first time, it demonstrated that human patients could be completely supported by a totally artificial device.
While this is not the end of the story, it's a very important milestone paragraph in the story, and my heartiest congratulations, guys.
They're much smaller, they're much more robust, and they work really beautifully to support a weak left heart, but no one had ever used one to replace a heart or used two of them to replace both sides of the heart. So we realized there was going to be some arts and crafts involved. We would actually take the Dacron, put it over the form, and then wrap it in fiberglass tape and silicone. And originally we were using stuff from Home Depot, you know, tile caulk from the bathroom department and drywall tape.
And the beauty of these continuous flow pumps, there's really two things: the obvious size, but they don't wear out. We haven't had one to wear out, so they behave like the natural heart, which is one of the reasons why, to me, this is the only hope long-term for a really totally implantable artificial heart that we can take off the shelf and spare patients like Mr. Lewis premature death.
So this is a milestone in cardiovascular history, in our opinion. It's a great step forward, and it's a great tribute to Dr. Cooley and Dr. Frazier and Dr. Cohn and their teams. Let me just read a statement from the family of Mr. Craig Lewis. This is the patient, and they asked that we read this here today. This is a quote. "We feel very blessed that the doctors at the Texas Heart Institute at St. Luke's Episcopal Hospital, the whole team, are able to use their God-given talents to continue to help people like Craig in this situation. We hope he continues to get better. Thank you."
So, as a neurosurgeon, I have a different perspective on central nervous system disorders, and in particular spinal cord injury, that I've acquired through years of practice, and now understanding the challenges of translating discoveries, such as what Dr. Anderson has done, into clinical testing, and then hopefully one day something that will benefit a patient. And that perspective has really informed me, and I think this is an appropriate setting to make this comment, that it takes a village, to use a phrase. It takes basic scientists, it takes translational experts, it takes industry, it takes academic institutions, it takes funding agencies such as CIRM, it takes regulatory bodies, ethics committees, and advocacy groups such as yourself to move all this forward. We all have a role to play, and when we come together and we realize what the common goal is, which is to create a therapy, to create a treatment for some of the most complex disorders that we can think of, we will benefit from all understanding that there's a role to be played in to advance all this. The other way I think about it clinically is that what we can't do in the brain, spinal cord, and eye is do an organ transplantation. Now, nothing fascinates a neurosurgeon more than the thought of a brain transplant, but we can't do that. But if we look at what is the proof of concept of transplantation as a therapeutic, you can look here on this list and you can see, excuse me, that on the left side, we started with kidney transplantation in the mid-1950s. And if you look at the history of solid organ transplantation and bone marrow transplantation and how sophisticated that has become, and what a very successful form of treatment for very challenging diseases, you can think that, well, that we can get some comfort that if we can achieve that kind of effect, not based on a whole brain or a whole spinal cord, but the constituents of a central nervous system, the cells, which is what Dr. Anderson is talking about, that maybe we can achieve some kind of clinical effect as well. So knowing that we can do this for solid organs, and again, it got off to a rocky start, I think, if you look back at the initial history, but it's evolved and it's continued, and they have persisted despite all of that because they believe this is one of the ways to treat those diseases. There's a list of central nervous system disorders, all of which have been on under consideration for how we might approach these from a cellular transplant perspective. So the cell that we're talking about today is the human central nervous system stem cell, or a neurostem cell. And Dr. Anderson has given you a very good primer on this cell and in general cell populations that you can think about that might be applicable. The key things about stem cells that make basic scientists and others interested is that they have two important attributes among many, and that is they have the potential for self-renewal. So you can put in a population of stem cells, and because there's a smaller population within that will self-renewal, you could, through a one-time transplantation, create something of a durable effect. In other words, the cells will survive, and they'll fall under the regulatory control of the host, the patient. The other aspect is that these cells have multilineage potential, so they can become the building blocks of the central nervous system. Those building blocks, neurons, oligodendrocytes, and astrocytes, are all important for normal function, but they're also damaged in disease and injury. And so if you can think about augmenting the function of those cells or replacing those cells through transplantation, then you start to think that you have the sound footings for a therapeutic, a potential therapeutic. So there's been a number of trials that have shown the way that we can transplant tissue into the brain, into the central nervous system safely. We know we have feasibility, tolerability, all important clinical milestones to achieve as you think about some of these things. Spinal cord injury on this list is something that has captured all of our imagination for a long time, because if we can reconstitute function over a few inches, a few centimeters of the central nervous system, a very critical part of the central nervous system, you could achieve a benefit. So the scale of what we have to correct is different than perhaps other disorders of the central nervous system that are listed here. And because these are injuries that occur in young people, I think even more so, we're dealing with patients who face a lifetime of disability. And if we can create benefits, perhaps not at the very beginning, better locomotor control, but other things as we advance in terms of cellular transplantation, that we will have done some good. So the company that I work at is a publicly traded biotech company. It's small, but we've tried to be productive in the last few years, and we've been able to do that based upon academic collaborations, regulatory interactions, ethics interactions, and so forth, to get the basic science data, like it's been done at UC Irvine, and to think about now, how do we get into the human study? How do we begin to ask the questions in the human setting? And we all know that stem cells is a very provocative subject, and it's a hurdle to get these first studies underway. And what you see here is a list of the trials that the company has either completed or is ongoing, and I'll talk about them very briefly, and then I'll talk about the spinal cord injury trial and our interim results. So the first one was the first in human transplantation authorized by the FDA of a neurostem cell into the brain. And it was for a disease, a lysosomal storage disease, Batten's disease, and this was started in 2006. And we put up to 1 billion cells into the brain of children with a fatal lysosomal storage disease. And from that study, we learned that we could do that kind of dose. We could do it surgically. The children would tolerate the surgery. They'd tolerate the immunosuppression for the short time after surgery. And then, because this was a fatal disease, we knew that there would be opportunities to look at the reaction of the brain, which is a very critical, bittersweet aspect of some of the research that we do. And the 3 children that have come so far in the trial, their families allowed us to do postmortem examinations of the brain. And what we learned, in addition to safety and tolerability, was that the stem cells would survive. And this was a critical thing to begin to establish. And this just shows that we could find the cells not only where we put them into the brain, but we could find that they had migrated to deeper parts of the brain. So this is starting to replicate some of the data that we see in the animal models, and that the cells persisted after we had the planned stop of immunosuppression. So as you think about cell therapy, it's like building a house, and the first is the foundation, and a lot of that foundation is safety. That allows you to the next step to begin to ask the more complex questions. And this is what we got through this first trial, and one of the outcomes was understanding that the cells look like they survive. The next trial that was done, which is the second IND authorized by the FDA for transplantation of neural cells into the brain, was for Pelizaeus-Merzbacher disease. And I won't go into too much detail, but this is another fatal myelination disorder that occurs in children. In fact, sometimes you can diagnose this within hours of birth. Our concept here was to take the neural stem cells, which we recognize from basic science work, that will become oligodendrocytes and then myelinate. Well, this disease is a hypomyelination disorder. And our goal here was to not only continue testing safety, but ask whether we could detect signs of myelination, detect a biological effect of the cell. And this work was just recently published when we completed the trial this year in Science Translational Medicine. And what you see here is a graph of an MRI technique in which the cells in the area of interest, the region of interest in the brain, is in red, and the other aspects of the brain are in blue. And what we see by this MRI technique is that in all 4 of the patients in this trial, the regions that we transplanted, we can detect signs of myelination by MRI. So again, another brick on the foundation of, if you will, of building this particular approach, which is, can we see signs of a biological effect? And we think we now have that with the PMD trial, and we're going to be moving on to a second trial in PMD. But this is particularly applicable because we know that myelination, an insult to the myelin, is part of spinal cord injury. So seeing that we can have this potential effect in the brain helps us think about spinal cord injury as well. So now the spinal cord injury trial. This is a phase 1/2 trial being conducted at the University of Zurich. Oh, excuse me. It's an open label trial, which means there's no blinding in this particular study. Everyone knows that you're getting the cells, the patients and the clinicians. And it's a single arm, so we don't have control patients at all. We have natural history data, which is pretty good in spinal cord injury that we can compare to. We've put a total dose of 20 million cells into the spinal cord, essentially identical to the paradigm that Dr. Anderson has developed with regard to the mouse. There was no reason to depart from that. And then we immunosuppress the patients for 9 months. And we immunosuppress because it's an allogeneic transplant, so it's one human tissue into another human. That requires some degree of immunosuppression, but we understand the central nervous system has a degree of immunoprivilege, so we don't think we need to do the immunosuppression indefinitely. And so it's a 9-month course to help protect the cells at least during the time when they might be most vulnerable to the immune surveillance by the body. It's 12 patients from T2 to T11. And the timing of injury here, we could go into a lot of details about this, but again, this was based upon the basic science work led by the team at UC Irvine and Dr. Uchida of when do we do the transplant. And in this particular trial, looking at that preclinical data, we decided that the window was 3 to 12 months. So late subacute, early chronic, however you'd like to term that. But more importantly in this trial, what we felt was important was to move beyond just the patients who had complete loss of sensory and complete loss of motor function, because we can all agree that perhaps is the most biological, challenging template in which to try to measure an effect, even a modest effect, and incorporate within that trial, after we establish safety in the Asia A patients, the completely injured patients, to B and C. And that's what this trial will consist of, with 3 Asia A's that we've now dosed, and I'll be speaking to, onto Asia B, 4 of whom, and then moving into patients who are classified as Asia C. We have a data monitoring committee, and of course we have the typical clinical parameters that we look at in terms of the Asia sensory motor scores. But Dr. Kurt's team in the University of Zurich is particularly good at quantitative sensory perception tests, which are very refined ways of looking at segmental thoracic function in terms of heat and electrical stimulation, as well as evoked potentials. And when you're dosing the thoracic cord, you don't quite have the fidelity of looking at motor function like you do in cervical transplants. But we still have the obligation of understanding whether we've changed segmental function, either good or bad. And that's part of the reasons that we did the trial at the University of Zurich. So this gets into some of the details of the sensory, what I'll be taking you through. There's the light touch, which we're probably all familiar with. It's a very coarse measure. There's normal, impaired, or altered, and absent. But the field is starting to evolve more sophisticated ways of looking at segmental thoracic function, and that is the quantitative testing for heat or electrical stimulation, in which those stimuli can be graded and become a more objective measure of outcome, both by segment as well as looking at evoked potentials from those stimuli. So these are the Asia A cohort patients. There were 3 in this cohort. They range from 23 to 53 years of age, and they were all dosed in last year, 2011. They had a T8, T9, and T4 lesion, and they are generally between 4 and 7, 8 months after injury. The cells are introduced into the spinal cord through a typical standard neurosurgical operation where you expose the spinal cord here, open the dura, and this was the first patient in the trial. And what's remarkable is that when you look at the MRI of this particular patient, you would have thought that you would see much more evidence of external trauma to the cord. And when we look and expose the cord, we see that there's more continuity than what you might have thought anatomically, which is good from a transplant standpoint. The cells are introduced 2 locations above and 2 locations at the margin of the lesion, not directly into the traumatic site, but at the margins. Again, based on the preclinical glimpses of work that we have with Dr. Uchida and Dr. Anderson. And the surgeon simply applies a very small needle, a very small cell suspension in each one of those locations through an open technique. So we look at the patients at different intervals at 3, 6, 9, and 12 months. And we had a DMC review of the data at 4 months that tells us whether we think we have ongoing safety in which we can move and continue with the trial. So at the end of 6 months, we don't have any adverse effects that we think are related to the cells. There's no evidence of increased pain thresholds or abnormal pain thresholds. And the surgery and immunosuppression, which are not insignificant for a spinal cord injured patient, have been well tolerated, and those look feasible so far. We spend a lot of time examining the patients very closely, not only to detect a safety concern, but whether we think we're picking something else that might be a signal of a biological effect. And what we've seen now at 6 months is that in 2 of the patients, we've seen gains in their sensory levels, and 1 patient remains stable. We think these sensory gains are somewhat unanticipated and unexpected if you look at the natural history, and they're backed up by some electrophysiology data that Dr. Kurt and his team is also getting. So this is, I'll start to go through the data in the instance of time. Am I doing okay? Okay. This is the first patient in the study, a 23-year-old male who was an Asia A, T8. And you can see the classical instrumentation, and then this is the MRI. And you can see here the area of injury. So it's interesting, as I pointed out, that I expected as a neurosurgeon to see a much more traumatized external part of the cord when we exposed this, because you can see there's absence of tissue, certainly within the central portion of the cord. But it gets to confirming what we all know, or think we know, which is that there's a rim of tissue, even in the most severely injured patients, that may be preserved enough and amenable to the effects of a transplantation. So in this first patient, we looked at all the different parameters that I just mentioned, and he's stable. He doesn't report any changes in sensory levels, and this just shows the Asia light touch examination pre-transplant and at 6 months. So essentially no change. We know that there's a margin of transition between segments that feel completely normal and then segments in which there's no sensation, and then there's this in-between margin, which in him essentially is stable. So subject 2 had this pattern of light touch. That was a little twitchy. So at 6 months, you can see that there's been a change in his light touch examination. And what Dr. Kurt and his team determined with this patient is that he didn't lose light touch perception per se, but he reported that this light touch was more robust, more vivid. And to capture that with the light touch examination by the Asia, Dr. Kurt remained true to it and says, well, it's different, and we'll score it as sort of the yellow, the impaired, if you will, which is not precisely accurate. But then what was good is that we had these other backup tests to determine what might be going on. So when you look at the patient's ability to understand electrical perception, this was pre-transplant. Now, post-transplant at 6 months, it's starting to normalize the electrical perception threshold, its ability to detect electrical stimulus. So now what we saw by the light touch examination is starting to be backed up by one of these other quantitative sensory measures, which is he's reporting an increased ability in segments below the injury of increased electrical perception. Well, another sensory modality in addition to light touch and electrical is thermal or heat. And Dr. Kurt and his team have a similar test, a quantitative test for that. And this shows that pre-transplant, he had this area of diminished area in the segment. I can't quite make that out, but it's below his level of injury. And then at 6 months, again, this is now normalized. So what he reported in his Asia touch examination was a vividness, a robustness that they felt needed to be captured. But when you look at his electrical and heat perceptions, you can see that they're starting to normalize. So all of that is a good sign that certainly we're not doing any harm, and potentially a sign that maybe something is happening biologically. So when you, in addition to asking the patient about what they perceive, which is the perceptual aspect, you can do evoked potentials. And what you see over time here with the heat is that pre-transplant in 3 months, it's fairly flat as I understand this examination, but as the time goes on, you start to see we're picking up amplitudes and latencies, and this is a very, very objective test, the electrophysiology. This is the third subject in the trial, 45 years old, T4 injury, high on the thoracic cord. And again, you see the typical changes in the cord, if you can make them out on the MRI here, of what you'd expect to see, particularly with a patient with a complete injury. He is 4 months after injury when he was transplanted. And pre-transplant, he has multiple segments below in which he has no light touch perception whatsoever. At 6 months, he's reporting an increase in light touch perception that's probably 5 or 6 segments below his initial T4 area of injury. When you add to that the electrical perception, so a way of starting to confirm what he's reporting, is that we also see a transition from essentially no ability to detect electrical perception and now starting to pick up some of those thresholds. Now, when you add in the electrophysiology, which is the SSEP, you can see that at T4, things are very flat pre-transplant, but that at 6 months, we're starting to see amplitudes and latencies, which start in reflective of a signal that's passing through the injured area up into the brain. When you look at T7, which is several segments below his level of injury, again, you see somewhat flat, essentially absent SSEP transplant. And now at 6 months, again, we're starting to see deflections in the signal, amplitudes and latencies that's starting to show that some electrical signal is passing from below up into the brain for them to detect by the electrophysiology measures. So this is the 6-month data for the 3 patients in the trial. We have no safety concerns, as I've mentioned so far, which is good as we move forward. But what do we think about the biological things that we're starting to see? And there's always an interest in over-interpreting open-label phase 1 data, and we have to be careful about that. But there's a risk in under-interpreting as well. One of the ways that you address that is try to look at what we know about normal sensory changes in patients who have these type of injuries. And this is data that comes from an article written in 2011 about what do you expect to see in terms of segmental changes at this time window with this type of injury. And what you see is most patients don't change at all. They don't have a 0 change in their segments. But a few patients, very small percentages, probably 1%, will get gains between 4 and perhaps more than 6 segments at that time. If you plot out where our first 3 subjects are appearing, they're falling on this distribution, but 2 of them look like they're toward the end of the distribution. So that helps put into context how we interpret this data. So from our perspective right now, the trial is continuing. The DMC is obviously understands that we can move into the Asia B cohort now, and they're continuing to monitor the trial. We feel good about the safety profile that we have, and we're very intrigued and comforted and enthusiastic about some of these signals that we're starting to see biologically. The fact that they're persisting now is even more encouraging. The trial is going to be open to patients who now have incomplete injury, not only to European patients, but patients within the US as well and Canada. So I'll end with this slide. I'm sure that when we look back in 10, 20, or 30 years, some of our efforts now will seem very crude, but I don't think we'll be faulted for having started to explore one technique, a tool that maybe we can offer to repair the central nervous system. And this is not unlike other tools that we're going to be developing, that there'll be cells, there'll be other ways of addressing human disease. But I think it's important that for all the challenges that we face, that we've started to ask these questions. And with each trial that we do, we'll learn more about the disease and learn more about the potential intervention. So thank you very much, and I think we're going to take questions. Thank you.
Brock, now we've got the data to prove it.
So should we have a look at your baby? You're 27 weeks now, aren't you?
Yeah.
Okay. Listen to baby's heart beating. Perhaps a little bit. Oh, that's a nice one, isn't it? It's always good, isn't it?
Birth defects represent one of the major cause of morbidity and mortality in children. And there's about 3 every 100 baby which are born with a major birth defect. We are facing many times with a situation in which we don't know what to do for these babies.
It's a girl. Okay. I collaborate with Paolo Di Coppi. I look after the mums while they're pregnant. Paolo visits the fetal medicine unit and he sees the mums if the baby has an abnormality because he looks after the baby when they're born. And we're trying to develop treatments using stem cells in the amniotic fluid around the baby.
The stem cells have represented a major breakthrough because it's the possibility of growing cells outside the body to make a repair to the children once the cells have been built in the laboratory. So what we have developed in the last few years, and this has been initially the work we have done with Anthony Atala at the Wake Forest Institute of Regenerative Medicine, and is the possibility of deriving stem cells from the amniotic fluid. These are not embryonic stem cells, but are neither adult stem cells. They have characteristics that are in between the two cell types, and for that
made with a person's own cells, eliminating many complicated aspects associated with transplants, artificial organs, etc. These scientists are the members of the Tlisti Lab at the University of California in San Francisco, and their findings were published in their paper titled Rare Somatic Cells from Human Breast Tissue Exhibit Extensive Lineage Plasticity, published in the Proceedings of the National Academy of Sciences in March 2013. These scientists were studying normal human breast tissue and were interested in seeing if this tissue contained cells that could produce a variety of different cells from different tissues. To tackle this question, they looked at markers that are located on the surface of the cell that they found to be different between cells that had the ability to produce cells of different organs compared to cells that did not have that ability, but instead could only produce one type of cell. They found that 2 cell markers were different between these 2 types of cells. One marker was increased, CD73, and one marker was decreased, CD90, in cells that could produce cells of different tissues compared to cells that could not. This finding gave the scientists the ability to go on a fishing expedition to find cells in normal human breasts that had lots of CD73 and very little CD90. And surprisingly, they found some. Let's color these newly discovered cells in red from here on. They isolated these red cells and wanted to examine them a little closer. To start off, the scientists found that these cells contain a lot of the same proteins as embryonic stem cells. So they wondered if they were also able to form a variety of different tissues, like embryonic stem cells. So first, the scientists wanted to see if these red cells could produce cells of the breast tissue, where they were discovered. And indeed, when a group of red cells were allowed to divide in the presence of very specific conditions,
can be corrected before birth, we can use his own cells for a treatment. So we can culture his or her cells outside the womb and expand them and eventually correct, for example, a gene that is altered or missing using gene therapy technique, and we can inject back the cells into the fetus before birth, and so somehow improving his option of life once the baby is born.
As humans, we develop from a group of cells called embryonic stem cells. These cells can make up all of the tissues in our bodies. But once we are born, these embryonic stem cells are long gone. In place of these cells, our tissues have their own specialized stem cells that can repair that specific tissue in case of injury or to replace cells that are worn out. But they can only produce cells of that tissue and no other tissue. For example, a muscle stem cell can only produce a muscle cell, not a heart cell. That's the job of the cardiac stem cells. The scientists of this paper wondered if there may be some cells hiding out somewhere after birth that can make a whole range of tissues, like embryonic stem cells. This would be a very interesting find because of the ethical issues associated with studying and culturing human embryonic stem cells. But if there existed cells that resembled embryonic stem cells that could produce a variety of different tissues, it would open up many opportunities for regenerating entire organs.
We talk about the fact that we all have stem cells in every organ or tissue, and it's true, we do. But one thing that very few people talk about is the fact that And one of the things that we're working on is the fact that as we age, we lose some of our stem cells. So I actually believe that aging is a failure of stem cells. What do I mean by that? What I mean is that as we get older, not only do the number of cells that we have that can repair our body go down, but the function, the ability of the ones we have goes down as well. You know when you're 2 and you fall down and you scrape your knee? You heal and you don't have a scar for the rest of your life. When you're 62 and you fall down and you scrape your knee, you're probably going to have a scar. And that's because as we age, we lose stem cells, cells that our body uses to repair every organ or tissue. The cool thing is, and not many people are looking at this, is there's a difference in how that happens in men and women. So one of the things that I'm thrilled about is the chance to work with Stephanie Coulter at the Women's Heart Health Program here, along with the colleagues I'm already working with around the country, to begin to define the differences in how men and women repair their bodies, how our stem cells work, and how they differ in men and women. You know, we know women
represent a big advantage for therapy. These cells would represent an ideal source for building organ and tissue that are missing in the fetus. And this is because we can predict and diagnose very accurately these diseases before the fetus is born. Once this diagnosis is made, however, we have about 20 weeks of gestation in which we can plan the engineered organ to be built outside the baby, that then can be eventually implanted once the baby is born. So we can correct that defect at birth using its own cells. Here is where we do receive the samples from Anna's. Once the cells are isolated from the amniotic fluid, they can be easily expanded in the incubator and eventually engineer in a three-dimensional structure that can mimic the organs that the baby is missing. These organs can be expanded and grow in these bioreactors.
We haven't got any treatments that will actually work yet, but we're working on this whole area. For instance, the mum that we scanned earlier on has got a completely healthy, normal baby as far as I can tell on scan, and we're not looking to treat anybody right now, but we are trying to develop new treatments which will improve already existing treatments or to develop completely new therapies using the stem cells that are in the amniotic fluid.
In other situations in which we can intervene even before birth, we can use the same cells to treat the fetus. So if we know that the fetus has some malformation that